Hello everyone, welcome back to Basic and Applied Microbiology classes. I hope you all are doing good. So today we are going to take 50 MCQs on history of microbiology. Let's start. See the first question. Who is known as the father of microbiology? Aristotle, Edward Jenner, Antony von Leeuwenhoek, Robert Koch. So the correct answer is Antony von Leeuwenhoek. Antony von Leeuwenhoek has made significant contributions in the field of microbiology. During his lifespan, he constructed around 250 simple microscopes, and using his own microscopes, he examined diverse range of samples including human blood, his own tooth scrapings, plant and plant materials, lake and pond water, and observed the presence of different type of microorganisms including bacteria and protozoa. That's why Leeuwenhoek is also known as father of bacteriology and protozoology. Let's take next question. Who performed the Swan-Neck experiment to resolve the controversy of spontaneous generation versus biogenesis? So, firstly, you should know about it that during Antony von Leeuwenhoek's time, whatever findings were made, that was actually the part of discovery era. And after discovery era, there was a period which is known as transition period. During that period, actually there were two groups of scientists, we can say. One group was of belief in spontaneous generation, means they were of this belief that living organisms could develop from non-living material. On the other hand, another group of scientists was of this belief that always living organisms could develop from living organisms biogenesis. So to resolve the controversy between these two group of scientists, or we can say between spontaneous generation and biogenesis, one experiment was performed which is known as Swan-Neck experiment by a scientist, which has led to the end of this controversy, right? So let's see who was the scientist who performed this experiment. Francesco Redi, John Needham, Spallanzani, Louis Pasteur. So the correct answer for this is Louis Pasteur. And here I have also included one picture to show you the Swan-Neck experiment. Why this experiment has been named as Swan-Neck? Because to perform this experiment, Louis Pasteur has used special type of round bottom flask with its neck curved into the shape of a swan neck. Let's see next question. Who was the first person to observe and accurately describe microorganisms and called them as animalcules? Let's see the options. Robert Hooke, Antony von Leeuwenhoek, Roger Bacon, Kircher. So the correct answer for this is again Antony von Leeuwenhoek. Leeuwenhoek has constructed very simple microscopes, and this picture is showing you one of the finest piece of microscope constructed by Leeuwenhoek. And of course it was simple microscope as it included only the use of one lens, and whatever observations Leeuwenhoek has made using this microscope, which was constructed by himself from diverse range of samples like lake and pond water, plant and plant materials, etc. Those microorganisms drawings he has made very nicely in his time, and also sent the compilation of all these drawings of microorganisms showing rod shaped, small rods, large rods, and of course a spiral eye to the most popular scientific society of his time, that is Royal Society of London. To Royal Society of London, Leeuwenhoek in 1676 has submitted all his observations, and of course that's why 1676, the year in which Leeuwenhoek has made so good, has taken so good observation and accurately described microorganisms, that year also marked the birth of microbiology subject. And of course, why Leeuwenhoek has called them as animalcules? Because till 1676, nobody was aware about microorganisms, that there is also a field microbiology, and of course microorganisms are present. Everybody was known about biology only. So what Leeuwenhoek found, Leeuwenhoek when submitted his all the compilations, observations to Royal Society, he has used this term animalcules, means something I have observed in different samples that was looking like animals, but they are not actually animals. Later on, these animalcules were found or were called as microorganisms. Let's see the next question. Koch's postulates were published in which year? So we all know about Robert Koch's contribution. Robert Koch is a very well known microbiologist who has worked a lot in the field of germ theory of diseases. And you should know that Robert Koch has introduced 4 postulates to study the etiology of a disease. What is etiology? Means to find a relationship between a disease and its causal agent. If any disease is occurring, of course it is due to some causal agent. Which causal agent is related to a particular disease? In order to understand this etiology, Robert Koch laid down postulates. 4 postulates Robert Koch laid down. Let's see in which year he laid down these postulates. 1861, 1881, 1894, 1884. So the correct answer for this is 1884. In 1884, Robert Koch laid down his postulates that are used till date to establish the relationship between a disease and its causal agent. Let's see the next question. Who discovered first wonder drug penicillin in 1929? Let's take the options. S.A. Waxman, Flore and Chain, Alexander Fleming, Martin. Correct answer for this is Alexander Fleming, right? So here you should know it has a very interesting story associated with it. What? The discovery of penicillin or wonder drug. Once what happened, Alexander Fleming went for vacation, and when he came back from vacation, he observed a plate in his laboratory. What he observed, that the plate was of a bacterium, and there was a bigger colony, large size colony, which was of a fungi, and around that colony, near around that colony, there was no growth of bacteria. Okay, then he assumed, or he suspected from this, that this fungal colony is going to produce something which is going to be diffused through the agar medium, and it is not allowing near to it the growth of any kind of bacteria. Later on, this substance was purified and it has led to the discovery of penicillin. Why it is called as penicillin? Because this substance was actually purified from the colony of penicillium. This is a very interesting story behind the discovery of penicillin by Alexander Fleming. Let's see next question. Who performed 3-jar experiment on meat to disprove spontaneous generation theory in 1665? Right, let's see the options. Robert Koch, Franz Coolje, John Tyndall, Francesco Redi. So let's make your understanding clear. It is the same kind of question. As I have told you earlier, there was a controversy between spontaneous generation and biogenesis between two group of scientists. So what actually happened, many scientists came forward at that time to resolve that controversy. Of course, that has ended with the experiment Swan-Neck, which was performed by Pasteur. But one such experiment to disprove spontaneous generation theory was also performed by another scientist whose name was Francesco Redi. So Francesco Redi performed a 3-jar experiment, which also became very popular at that time. But of course, it was not that much accepted as Pasteur's experiment Swan-Neck was accepted at that time. So let's see next question now. Who is known as the father of antiseptic surgery? Let's see the options. Joseph Lister, Paul Ehrlich, Fanny Alchemius-Hesse, Robert Koch. So the correct answer for this is Joseph Lister. Joseph Lister actually introduced the use of carbolic acid during surgical dressings, and this was the picture of a kind of carbolic acid sprayer which was used by Joseph Lister in his time. Okay, that's why Sir Joseph Lister is known as father of antiseptic surgery. Let's take next question. Who were the first to introduce the idea of using cotton plug for plugging microbial culture tubes in 1854? The options now: Theodore Swan, George Schroeder, and Theodore von Dust, Louis Pasteur, Richard Petrie. So the correct answer for this is B, that is George Schroeder and Theodore von Dust, who proposed the idea of using cotton plug. Very important contribution by these scientists. Let's see the next question. Who proposed the use of agar in culture medium? We all are knowing that, of course, nutrient medium, when we use in microbiology laboratory, it is of liquid type, which is broth, and it is agar, which is solid medium, right? And of course, it's a product of Robert Koch lab. But let's see use of agar incorporation, or I can say incorporation of agar in culture medium was introduced by which scientist? Robert Koch, Antony von Leeuwenhoek, Phenylshemeous Haze, none of the above. So right answer for this is Phenylshemeous Haze. Now you might be thinking that previously I told that it is a product of Robert Koch lab. Yes, because Robert Koch in his time has worked extensively on development of bacteriological techniques, and there was one assistant of Robert Koch. His name was Walther Haze. Walther Haze was very much aware that it has become difficult in the lab to solidify a medium, a liquid medium. In order to solidify liquid medium, we are facing a lot of difficulty. Then at that time, that Koch's assistant Walter Haze discussed this problem with his wife Fanny Alchemius Haze, which is commonly known as Fanny Haze. Then Fanny Haze at that time was making the use of agar for solidifying jams and jellies, and she was very much aware that agar is actually not subjected to degradation by various type of microbial contaminants. Then she suggested her husband at that time that why not to use agar for solidifying a medium in your laboratory. And of course, as per the suggestion of Fanny Alchemius Haze, the use of agar was very much successful, and till date we are going to make the use of agar as a solidifying agent in microbial culture media. So let's take next question. Who developed Petri plate, Petri dish, a container for solid culture medium in 1887? So again, I must say it is again an outcome from Robert Koch's lab. Let's check the options now. Robert Hooke, Walther Haze, Chamberlain, Richard Petri. Then again, you should know here, Petri plate, we all are aware we are going to use as I have shown in this picture. And of course, in this way, we add media into this and then we get the colonies, and of course, we note down our results. But the Petri plate, this Petri plate actually was introduced by Richard Petri. Again, I have told you that Richard Petri, his full name is Julius Richard Petri. He was again one of Koch's assistant. Okay, Robert Koch's assistant. Okay, so again we can say that again the Petri plates what we are going to use they are used in pure culture techniques, or we can say as a bacteriological technique they are used. So they are again the outcome or contribution from Robert Koch's lab. And of course Julius Richard Petri was involved behind introduction of this Petri dish. Let's take next question. Who discovered Mycobacterium tuberculosis in 1882? Right. So Mycobacterium tuberculosis, we all are very well aware. It is a very fatal organism, and of course the causal agent of the disease, tuberculosis. Let's see who discovered it. Robert Koch, Eli Machnikov, Domack, Emile Rox. So the correct answer for this is Robert Koch. Robert Koch in 1882 discovered this organism. Let's see the next question. Fleming, Flore, and Chain shared the Nobel Prize in Medicine for the discovery and production of penicillin in which year? Right. So, of course, we all know that Fleming discovered what? Penicillin, right? Discovery of penicillin was done by Sir Alexander Fleming, and its commercial production, or we can say production on large scale, was actually worked out by Flore and Chain. So, combinedly, Fleming along with Flore and Chain received the Nobel Prize. They shared a Nobel Prize for the production of penicillin, of course, as well as for discovery. So let's see in which year they got this Nobel Prize. 1929, 1940, 1941 and 1945. So the correct answer for this is 1945. In 1945 they shared this Nobel Prize. Let's see next question. Name the German bacteriologist who proved that anthrax is caused by Bacillus anthracis in 1876. So of course the answer is very clear. From here actually the journey of this scientist began, and later on he worked out so many bacteriological techniques. So let's see the options. Pasteur, Chamberlain, von Plances and Koch. So the correct answer for this is Koch. Koch started his journey from anthrax to resolve the issue of anthrax, and from that he made so many contributions, of course, in isolating pure culture as well as bacteriological techniques. Let's see next question now. Who gave the concept of one gene, one enzyme hypothesis? Right? One gene can code for one enzyme. This hypothesis was proposed by some scientists. So let's see who was or who were those scientists. Jacob and Monod, Beadle and Totem, Zinder and Lederberg, both A and B. So the correct answer for this is Beadle and Totem. Beadle and Totem actually proposed the concept of one gene, one enzyme hypothesis. Mean, according to them, they proposed this, that one gene is responsible for coding one enzyme. Okay, and of course, in order to prove that, they performed an experiment, and in their experiment, they have used a red bread mold, or we can say a fungus named as Neurospora crassa. Neurospora crassa spores were used by Biddle and Totem to perform an experiment to prove one gene, one enzyme hypothesis, right? So let's see next question. Which of the following scientist was in favor of spontaneous generation theory? So already I have told you about spontaneous generation controversy versus biogenesis. So let's see who was the scientist in favor of that. Francesco Redi. So we all know no, it is he was not in favor. Why? Because he performed 3-jar experiment. Already we discussed. Louis Pasteur, no, he ended actually this controversy. He was not in favor. He performed Swarnack experiment. John Needham, and then Schroeder and Von Dust. So out of C and D, the correct answer is John Needham. John Needham performed actually an experiment in favor of spontaneous generation theory. What he told that I have taken a medium, a nutrient solution, right? In that nutrient solution, I am going to heat that nutrient solution. After that, he said, I am keeping it for some time. And of course, after some time, again, the growth will be there of microorganisms in that flask. So then by this experiment, what he tried to prove that if nutrient is already having some biological agents or living things, by heating they are going to be killed. And then of course now the medium is free of, nutrient solution is free of actually microorganism. After some time again microbial growth is there. It means non-living material, or we can say the nutrient ingredient present in the nutrient solution itself, they have the capability to give rise to microorganisms. Means spontaneous generation or spontaneous development of microorganisms is possible from the non-living nutrients present in the nutrient solution. Okay, no vital force is required. So of course he was in favor of spontaneous generation theory. And Schroeder and Von Dust, already I told you, they were actually involved behind the introduction of the cotton plug. So let's see the next question. Who was responsible for saving a principal industry of France involved in manufacturing of beer and wine? Right. So let's see the options. Liebig, Schulze, Emile Rox, Louis Pasteur. So the correct answer for this is actually Louis Pasteur. Louis Pasteur was actually a French chemist who was working in the University of Lille in France, right? So he actually worked a lot on saving a very popular and principal industry of France who was involved in manufacturing of beer and wine at that time. How actually he saved that industry? At that time, beer and wine was going to be spoiled again and again during manufacturing in that industry. So Louis Pasteur actually proposed a heating method which actually targeted the spoilage causing microorganisms, and of course saved this industry. And that heating method to target the spoilage causing microorganisms or disease causing microorganisms is very well popular nowadays as pasteurization. So pasteurization was actually proposed by Louis Pasteur firstly for beer and wine, and later on it became more popular, of course, with its application for milk, right? Whatever full cream toned milk, double toned we are nowadays procuring from the market, of course it is pasteurized one. Again, it's a contribution of Louis Pasteur. Let's take next question. Who worked on the development of pure culture techniques in microbiology? So already so many times I have told you who was the person behind development of all these culture techniques. Let's see the options and find the answer. Beizernik, A.B. Frank, Robert Koch, Joseph Lister. So the correct answer for this is of course Robert Koch. From Robert Koch's lab, almost a good number of requirements for isolating bacteria in pure culture, and of course for other bacteriological techniques have come up. So let's see the next question. Who was the first to treat smallpox with pus from cowpox lesions on May 14, 1796? Koch, Edward Jenner, Pasteur, and von Behring. Of course, first to treat smallpox with pus, and we can say treatment of smallpox or smallpox vaccination. This was first started by, of course, the father of immunology itself, who is known as Edward Jenner, right? Edward Jenner actually introduced the concept of vaccination, but at that time, no, this term was actually not used. This is a vaccine. Of course, he used it to treat cowpox lesions, to treat the smallpox one. So let's see next question. Who discovered streptomycin production from Streptomyces griseus in 1944? So let's see the option. Asser Waxman, Fleming, Loeffler, Buckner. So the right answer for this is after antibiotic penicillin discovery, a very much popular discovery was of streptomycin, and of course which was made by Asser Waxman. Salman A. Waxman made this discovery, and he is also known as father of antibiotics, right? Asser Waxman during his career screened around 10,000 or more than that bacterial actinomycetes strain in order to get good number of antibiotics. Let's see next question. Waxman received Nobel Prize in Medicine for the discovery of streptomycin to treat tuberculosis in which year? Of course, Waxman, for his notable contribution, he also has been awarded with the Nobel Prize. Let's see in which year he was awarded with this award. 1950, 1958, 1952, and 1953. So the correct option for this is option C, 1952, right? Let's see the next question. Fleming reported production of penicillin from which species of penicillium? So already I have shown you this picture, right, which is from the publication paper of Alexander Fleming itself, right? So in this picture you can see here this larger one is actually fungal colony, right? So this fungal colony was actually producing that substance which is nowadays known as penicillin, which was inhibiting the growth of what? Bacteria. So what was the species of this penicillium? Here the question is about that. So let's see the species. Aeruginosa, Chrysogenum, Notatum, Digitatum. So the correct answer for this is actually Fleming discovered penicillin production from Penicillium notatum. Notatum was the species of penicillium at that time. And you should also update your knowledge. Penicillin commercially is going to be produced from Penicillium chrysogenum. It is a more appropriate strain, or it gives more yield, right? So commercial production of penicillin is carried out from chrysogenum, but it was actually discovered from the Penicillium notatum. Let's see the next question. DNA is the genetic material and carried information during transformation in bacteria. It was demonstrated in 1944 by which scientists? Let's see the option. Beadle and Totem, Zinder and Lederberg, Oswald T. Avery, Colin M. MacLeod, Maclean McCarthy, and D. Griffith. So Griffith is not there. Why? Because Griffith has just performed an experiment using smooth and rough strains of bacteria, pneumococci, and he just found that some transforming principle is involved over there, which is going to convert the rough bacteria into the smooth bacteria. But at that time he was unaware which is actually the active molecule that is actually responsible for this, or which is actually the information carrying molecule. And later on this transforming principle was discovered by these three scientists, which are there in the option number C. You can see Avery, MacLeod, and McCarthy. These three scientists actually performed an experiment very successfully, who led to the discovery of DNA, who proved it DNA is the genetic material or transforming principle, and it was also there in Griffith's experiment. Let's see next question. Dr. Hans Christian Gram developed gram staining in which year? So 1864, 1874, 1884, 1894. So the correct answer for this is option number C, 1884. We all know about gram staining. Gram staining is a technique which is used to classify bacteria into two types, gram positive and gram negative, right? So this gram staining method was actually developed and first used by Dr. Hans Christian Gram in 1884, right? Let's see the next question. Who found the antimicrobial activity and therapeutic potential of trypan red magic bullet against African sleeping sickness, right? So let's see, this is showing you something different from antibiotics. Antibiotics are actually produced by microorganisms, right? We can say they are the secondary metabolites produced by microorganisms. But here we are going to see the use of a dye named as trypan red against what? Against a disease, African sleeping sickness. So we should see here who was actually the scientist involved behind the introduction of this dye. Sakiro, Hatta, Domack, Fleming, Paul Ehrlich. Then of course the answer is what? D. Paul Ehrlich. Sir Paul Ehrlich also initiated, or we can say worked a lot on the development of chemotherapy, means the use of chemicals in order to treat various kind of diseases or to achieve their therapeutic benefits. Let's see next question. Who introduced the use of gelatin as a solidifying agent for solid culture media? Then already I shared with you previously that agar was the outcome of Robert Koch's lab, which was suggested by Fanny Haze. But of course, before agar, gelatin was actually also attempted for its use as a solidifying agent, although it was not successful. Why? I'll be telling you. Let's see the option first. Who introduced the use of gelatin? Pasteur, Lister, Koch, and Haze. So the correct answer for this is Koch. Robert Koch by himself first introduced the use of gelatin, and later on it was found that gelatin is actually a protein, and of course it can easily be degraded by proteolytic bacteria. Whatever number of microorganisms, bacteria, fungi, they can produce proteases or have proteolytic properties, they can degrade gelatin. Therefore we cannot get intact colonies on gelatin incorporated medium, solidified plate, as we can get for agar. And second problem with gelatin was this, that as the Koch was going to incubate his plate at temperature more than 25 degrees Celsius, then gelatin was going to melted out. Why? Because gelatin is having a melting temperature between 5 to 28 degrees Celsius. At around 28 degrees Celsius, gelatin convert into a liquid form. It will not be retaining its solidifying properties. On the other hand, if we talk about agar will still be acting as a solidifying agent at even higher temperature. And of course, we as a microbiologist generally need a temperature of 37 degrees Celsius in our laboratory. So gelatin was not working at that time very well. That's why it was actually replaced by the use of agar. Let's take next question. Who developed the technique of attenuating cultures? Attenuation, we all know attenuation is what? Attenuation is a very good principle which is used for development of vaccines, right? Attenuating means to remove the virulent properties, or we can say to remove virulence from a pathogenic microorganism. Okay, this is actually the principle behind the development of vaccines. So who actually developed this technique of attenuating cultures, and then so that they can further be used to treat the same kind of disease what actually they were causing initially, right? So let's see the options. Edward Jenner, Emile Rox and Martin, Koch and Emile Rox, Pasteur and Emile Rox. So the correct answer for this is D. Pasteur, along with his research associate Emile Rox, actually developed this technique of attenuating culture, which is still in use today for development of vaccines. Let's take next question. Who discovered tetanus, lockjaw, antitoxin in 1890? Right? So antitoxins, of course, you all also know that of course diseases are of many types, right? Some are intoxications and some are infections. When we talk about infections, they are caused by actually living organisms, and when we talk about intoxications, they are caused by ingestion or intake of toxins. So tetanus is a disease which is caused by toxins, and of course one antitoxin against tetanus was discovered in 1890 by which particular scientist? Let's check out. Emile von Behring, Shibasuboro Kitasto, both A and B, J. Martin. So the correct answer for this is von Behring and Kitasto. Option C is right here. Let's take next question. Who discovered diphtheria antitoxin in 1890? In the same way, diphtheria is also a kind of intoxication, right? Or we can say toxin-based disease. So let's see who discovered diphtheria antitoxin in 1890. Shibasuboro Kitasto, von Behring, J. Plances, none of the above. So the correct answer for this is von Behring. Von Behring actually discovered diphtheria antitoxin. Let's take next question. Who developed vaccine for anthrax in 1881? In 1881, anthrax vaccine. Always remember, anthrax was worked out a lot, and its causal agent was found and isolated in pure culture by Robert Koch, but of course vaccine was developed by whom? Let's check out that. Koch. No, Koch has actually worked on this particular disease and its causal agent, but not he was involved in vaccination. Emile Rox, Jenner, Pasteur. So the correct answer for this is Pasteur, D. Right? Jenner was involved in smallpox vaccination, which was actually the first vaccine introduced, right? That's why Jenner is also known as father of immunology. So let's check out next question here. Anthrax vaccine credit goes to Pasteur. Let's take next question. Who developed vaccine for rabies? So again, after the success of this anthrax vaccine, let's check out who was the person responsible for developing the vaccine for rabies. Pasteur, Jenner, Koch, Weizsäcker. So the correct answer again is Pasteur. Pasteur made a very important second discovery in the field of vaccination, that was for rabies, right? Let's check out next question. Who proposed the third kingdom protista? Right? So initially what was there? Plantae and Animalia were known. One group was belonging to plants, right? Plant kingdom. Another was for animals, animal kingdom. But later on a third kingdom was proposed, which was named as protista. All the bacteria, fungi, protozoans were kept under this third kingdom, and the introduction of this third kingdom was very important, and of course it made the microbial study different from plants and animals. Let's check out who was that scientist who proposed the third kingdom. Carl and Woese, they have proposed actually 3-domain system, not protista. Ernest Haeckel, Whittaker proposed 5-kingdom system of classification. Baltimore, Tamin and Baltimore, they made some other important discovery in molecular biology, restriction endonucleases, right? So the correct answer here is Ernest Haeckel. Ernest Haeckel is the right answer for, he was actually the person who was responsible for introducing this very important kingdom, protista. Let's check out next question. Who discovered endospores? Endospores, we all know, they are the dormant form of bacteria which are produced under unfavorable conditions of nutrient starvation, extreme of temperature, pressure, desiccation, etc. So see who was actually the person behind the discovery of endospores. Theodore Escherick, Spallanzani, Ferdinand Cohen, both B and C. So the correct answer for this is actually Ferdinand Cohen. Sir Ferdinand Cohen discovered endospores, right? Let's see next question. Application of Koch postulate is not feasible for which of the below given microbial group? Of course, we know about Koch postulate. Koch postulates were actually introduced to establish the relationship between a disease and its causal agent, right? But till date, we use Koch postulates. We isolate bacteria from a diseased host and we culture them in the form of a pure culture, and then we check their characteristics, and then we correlate whether actually they are the cause of that particular disease or not. But remember, Koch postulates are not applicable to some microbial group. Let's see what are those microbial groups. Viruses, Mycobacterium leprae, Bacillus, both A and B. So the correct answer for this is D, option D, both A and B. Means viruses and Mycobacterium leprae. Why Koch postulates are not possible for this? Because we should know that viruses and Mycobacterium leprae, they are obligate intracellular parasites. Means they are strict parasites. They required only host to grow, to cultivate. Okay, they cannot be isolated in the pure culture in the laboratory, so that we can further work on identifying them and relating them with a particular disease, right? So of course Koch postulates usage is also restricted to some organisms, but of course it is not applicable to all organisms, especially obligate parasites. Let's see next question. Who developed enrichment culture technique? Right? Enrichment culture technique, again, it is to favor the growth of a special group of microorganisms, right? So only one type of microorganisms we will enrich the growth of. We will create such kind of conditions in the medium that only one type of microbial growth should be favored, right? So that is known as enrichment, enriching a particular culture. So that enrichment culture technique has been worked out or developed by which particular scientist? So let's check out. Vinogradovsky, Weizsäcker, both A and B, Hellriegel and Wilfarth. So the correct answer for this is A and B. These both scientists, Vinogradovsky and Weizsäcker, has actually worked out these techniques independently. Both worked on the development of these techniques, but not in collaboration. Independently they worked out. This is the picture of Sir Vinogradovsky and this is the picture of Weizsäcker. Sir Vinogradovsky is also known as father of soil microbiology, and both of these scientists are very well known founder of soil and agricultural microbiology. Let's see next question. Who introduced the concept of fractional sterilization or intermittent heating to kill endospores? Okay, so to target endospores, one technique was introduced which is known as fractional sterilization or intermittent heating. Means periodic heating is done, heating and then incubation for some time. After that, again subjected to heating, we will subject the sample to heating and again incubation. This will allow what? If spores are there in a sample, they will germinate and they will convert into vegetative cell. In second heating step, those vegetative cells will be killed. So this is known as fractional sterilization or intermittent heating. And third name is also there, what I'll be telling you after options, because through that you will be able to get the answer. John Tyndall, Louis Pasteur, Spallanzani, Leeuwenhoek. So the correct answer for this is actually option number A, John Tyndall. John Tyndall, and this process of fractional sterilization, intermittent heating, is also known as Tyndallization. Tyndallization with the name of John Tyndall. Let's take next question. Who was the first to observe cells in 1665, right? First cells were observed by whom? Let's check out Zacharias Janssen, Galileo, Antony von Leeuwenhoek, Robert Hooke. So the correct answer for this is actually option number D, Robert Hooke. Robert Hooke has constructed his own microscope, which was a type of compound microscope. Of course, we can say the old version of microscope what we are going to use today. So here two lenses actually he used in that microscope. That's why it is called as compound microscope. One lens was eyepiece lens, or we can say ocular lens, and another lens was objective lens. In same type of lens we also can see in our laboratory in modern microscopy, right? What we use today, bright field microscopy. So this type of compound microscope was actually used by Robert Hooke to observe the cells in which year? 1665. So you can see many hidden questions are there. Who observed the cell? Robert Hooke, right? In which year? 1665. Second question. Which type of microscopes he has used? Compound microscopes, right? So what type of cells actually, in which sample he has observed the presence of cells? So they were Koch cells, right? So you can see and of course whom first time observed, then of course the answer is same, cells Hooke, Robert Hooke, right? So in this way you can prepare yourself for the exam or for the particular target what you are focusing on. Let's see next question. Who developed transmission electron microscopy? Transmission electron microscopy, of course, it's a type of electron microscope. Electron microscope, we know they are mainly of two types: scanning electron microscope and transmission electron microscope. Transmission electron microscopy has actually been used to study the ultrastructural analysis of a particular microorganism, right? So let's see who the options: Kerry Mullis, Ruska, Tamin and Baltimore, Watson and Crick. So the correct answer for this is Ruska. Ruska was actually a person from physics background, right? He developed this technique, transmission electron microscopy. Let's see next question. Griffith performed his experiment on bacterial transformation in which year? So we all are familiar with this experiment. Of course, with this experiment, of course, the journey of our biology and microbiology always get a kick, right? So let's see in which year this experiment was performed by Griffith on R and S strain to for bacterial transformation: 1908, 1918, 1928 and 1938. So the correct answer for this is actually 1928, right? Let's see next question. Who crystallized viruses for the first time in 1935? In 1935, viruses were first crystallized by whom? Let's see the options: Stanley, Ivanovsky, D. Harale, Hershey and Chase. So the correct answer for this is A. Option A, Stanley. Stanley first crystallized viruses. Okay. Let's see the next question. Who discovered conjugation? Okay, conjugation is what actually you know. With this picture, you are very much clear. Transfer of genetic material from one bacterial cell to another bacterial cell with the help of a sex pilus. This is called as the conjugation. So let's see who actually discovered conjugation. J. Lederberg and E. L. Totem, Avery MacLeod and McCarthy, Zinder and Lederberg, Frederick Sanger. So the correct answer for this is J. Lederberg and E. L. Totem were the scientists who discovered conjugation. Let's see next question. Who discovered transduction? What is transduction? Transfer of genetic material in a bacterial cell with the help of a bacterial virus or bacteriophage. This is called as the transduction. Let's see who discovered transduction. Walter Gilbert, Frederick William Twott, Norton Zinder and Joshua Lederberg. Both A and C. Then correct answer for this is Norton Zinder and Joshua Lederberg. Let's see next question. The first edition of Burge's Manual for Bacterial Classification was published in which year? Right? In which year? Burge's Manual is widely accepted scheme of bacterial classification, right? You should know. And of course, in which year its first edition was published? Let's see. 1923, 24, 25 and 26. So the correct answer for this is 1923. In 1923, first edition of Burge's Manual came, right? Let's see. Question number 4. Who disproved spontaneous generation theory? Of course, spontaneous generation already we have discussed. Ferdinand Kohn, Louis Pasteur, John Needham, Theodore Swan. The correct answer is Louis Pasteur with the help of his Swan-Neck experiment, what we have earlier discussed. With this, spontaneous generation theory was disproved. Let's see next question. The engulfment of disease causing bacteria by WBC's macrophages was discovered by whom? Who was actually responsible for this discovery that disease causing bacteria are engulfed by WBCs and macrophages? Let's see the options. Max Cooper, J. Lister, Edward Jenner, Eli Machnikov. So the correct answer is Eli Machnikov. Always remember, Eli Machnikov is also known as father of phagocytic theory. And Eli Machnikov was also awarded for his discovery with Nobel Prize. Let's see next question. The concept of spontaneous generation is related to which of the following theory? Theory of biogenesis, theory of abiogenesis, theory of anaerogenesis, none of the above. So the correct answer for this is actually theory of abiogenesis. Means what spontaneous generation concept was actually stating. It was actually of this belief in spontaneous generation that non-living materials can give rise to living organisms, right? So it means it was a theory of abiogenesis. Let's see next question. Who discovered viruses infecting bacteria? Bacteriophages. Stanley, Twort, Ivanovsky, Bezernik. So the correct answer for this is option number B, Twort. Twort discovered bacteriophages or bacterial viruses for the very first time. Let's see next question. Who among the following is the most famous founder of virology? So let's see the options. Pasteur, Bezernik, Nicholas Appert, A.B. Frank. So the correct answer for this is Bezernik. Bezernik is a very well known founder of virology. He worked a lot in this field. And if we talk about Pasteur, Pasteur is known as father of modern microbiology here. Nicholas Appert is known as father of canning. He was actually the person who introduced the concept of canning or appertization in food microbiology. A.B. Frank discovered mycorrhizae, okay, which is used as biofertilizer in agricultural microbiology. Let's see next question. Name the gram-positive bacteria against which mold produced inhibitory zone that resulted in the discovery of penicillin, right? Again, here we are going to again see the same thing. This is again the question related to penicillin. Here we are talking about the bacteria. The bacterial growth, the growth of which was actually inhibited by the penicillium. Let's see the options. Streptococcus pyogenes, Staphylococcus aureus, Streptococcus lactis, Lactobacillus casei. So the correct answer is Staphylococcus aureus. Whatever growth was actually inhibited by the substance produced by penicillium or penicillin produced by penicillium, the colonies were at that time of Staphylococcus aureus, right? During the discovery time. Let's see next question. Which of the following scientists carried out experiment in a specially designed box to prove dust carried the germs? So here we are going to talk about a dust-free chamber, or we can say dust-free device, which was constructed by a scientist, and with the help of that he proved that dust, or we can say dust present in the air, is the source of microorganisms. Non-living material, non-living nutrients or ingredients present in the broth cannot give rise to any living organism. So let's see the options. Louis Pasteur, T. von Dust, John Tyndall, Spallanzani. So the correct answer for this is John Tyndall. Sir John Tyndall actually devised this kind of dust-free chamber, and with the help of that he proved what? That dust carry germ with it, right? Let's see next question. All of the below given investigators have been awarded with the Nobel Prizes for their research contributions in microbiology, except. So see, it's a very important question. Who was actually the one who unfortunately didn't get any Nobel Prize for his contributions? Eli Machnikov got the Nobel Prize. Alexander Fleming was also awarded with Nobel Prize. Robert Koch was also awarded with Nobel Prize. So here is the scientist, or we can say he is also known as father of modern microbiology, Louis Pasteur, who has made many contributions in the field of microbiology, but he was not awarded with Nobel Prize. So now a question can come in your mind, why he was not credited for his contributions? Then you should know that Louis Pasteur passed away in 1895, and the Nobel Prize, awarding Nobel Prize, this started in 1901. So that is the reason that Pasteur has not received any Nobel Prize for his contributions. So thank you so much. I hope this information will really be useful for you. Wish you all the very best for all kind of exams you are targeting.